Good morning, grade 8. Let's continue with the discussion. We are still in the middle of cell division and genetics. Alright? We are done discussing about mitosis. And again, what are the enzyme involved in the protein involved in the cell cycle? Okay, let's have this one. What enzyme is involved in the uh, movement or the moving on of cell from G1 to S or from S to G2? What enzyme again? Anybody?
The Psy what is it? CMD?
CMD? What's CMD, Rabi?
Psy Cyclos? What I forgot what it was, sir.
It is
But it was C started with C.
it is CDK. Okay,
CBDK.
okay, cyclin dependent kinase. Okay, it is an enzyme involved in letting the cell move forward from G1 to S or from S or synthesis phase to G2. And what inhibits now the moving on of what protein is involved in inhibiting the movement of this cell from moving on? What protein? It is the Rabi.
Wait, forgot, sir.
What protein? It is called the P53. Okay. If
Um,
mitosis is for the diploid number of chromosomes and the output or the number of daughter cells in mitosis is 2, let's have meiosis. What's with meiosis? If mitosis ensures that the daughter cells will receive the same amount, the same number of chromosomes as the parent or parent cell, another type of cell division ensures that the number of chromosomes is maintained among species by reducing the chromosome number by half or haploid in an organism's sex cells or gametes. This is to ensure the number of chromosomes is maintained. Okay? If the number of chromosomes is not maintained, tendency is there are some problems or genetic problems that may occur. That's the problem. Okay, let's move forward. If you have question, please raise it. Right. This type of cell division is called meiosis. While mitosis and meiosis share similarities, they are different in two aspects. Number one, recombination of genetic materials occur, which results in variations. And number two, four haploid cells are produced at the end of meiosis, or four daughter cells are actually created at the end of the meiosis, except for, again, I've discussed this one last meeting or yesterday, except for oogenesis. Because in oogenesis, or the production of egg, only one is being retained and the other disintegrate, or the three daughter cells disintegrate. Because we need only one egg cell in every cycle. But in sperm cells, on the other hand, the spermatogenesis, we have four, okay, daughter cells, haploid daughter cells in every cycle. Let's move forward. Meiosis can be called gametogenesis, or when you say genesis, it is the formation. Okay, gametogenesis because it is responsible for the formation of gametes. Okay, the egg and the sperm. The specific terms describe the production of sperm is called spermatogenesis, the one that I've been telling you a while ago. The formation of sperm is spermatogenesis, And the production of egg or formation of egg is called oogenesis. Okay, please take note of the terminologies. These are just the basic terminologies. Meiosis results in the formation of cells that are haploid. Haploid meaning half the number of chromosomes or half the number of entire chromosome. Like for example, in humans, how many chromosomes do we have? How many chromosomes do we have? Anybody? Are you still with me?
Ah, yes, sir.
How many chromosomes do we have in humans?
Uh,
We have how many chromosomes?
46.
46. Okay, that's correct. We have 46 chromosomes all in all. So, in meiosis, instead of having 46, we will have how many chromosomes? In the gametes, how many chromosomes? Instead of having 46, we have 23 chromosomes. Okay? As a result of meiosis. Okay? Unlike mitosis, meiosis involved two divisions which shall we call meiosis 1 and meiosis 2. Okay? We have two divisions under meiosis. Meiosis 1 and meiosis 2. Let's tackle about meiosis 1. Meiosis 1 is coined or termed as reduction division because we are reducing actually. Okay? Prophase 1 is pretty much extended in meiosis. Okay, we started with prophase 1. At this stage, chromatin condenses to become the shorter and thicker chromosomes. Homologous chromosomes, those that carry the same type of information pair with each other, lie side to side to form a tetrad. Because of this, because of prophase 1, we actually share different traits from our parents. We acquire those traits because of this tetrad. Okay? Because of the homologous chromosomes. It is during the formation of crossing over, this is now the crossing over, occurs. Crossing over between homologous chromosome allows shuffling or exchange of genetic material and produces variation in the offspring. This is the reason also why you have siblings that is not actually don't have siblings that is not related to your face, for example, or the color of your skin, because there is an exchange of genetic materials happen during this crossing over, right? But it doesn't necessarily mean that it is not your sibling. It is just it means to say that the shuffling or the crossing over is effective because you are now creating what? Variety or variations. Similar to mitosis, the nuclear envelope begins to break down, nucleus disappears, and centrioles move to the opposite poles. Same with mitosis. But meiosis is different because we are now exchanging genetic characteristics from the parents or from your parents. Okay? The second phase of meiosis 1 is the metaphase 2 or metaphase 1 rather. Homologous chromosomes move to the equatorial plate with their centrioles or centromeres attached to the spindle fibers and oriented to the opposite poles. So it is now start moving slowly, okay, towards the equatorial. Okay, and because of this, also there will be alignment of the chromosomes. Okay, again at metaphase 2, homologous chromosomes move to the equatorial plate. Remember that the metaphase The one that we can categorize metaphase is the moving on to the equatorial plate or in the equator, right? During anaphase 1, the third phase in meiosis 1, the homologous chromosomes separate and move towards the poles. It is now starting to move towards the poles. Unlike mitosis, sister chromatids remain together in anaphase 1. They are not actually being separated in um, anaphase 1. And the cell membrane begins to cleave. Okay. In telophase 1, the fourth phase in meiosis 1, the nuclear envelope begins to form around the chromosome, sister chromatids or sister chromatids, and eventually ends in cytokinesis, forming two daughter cells with half number of chromosomes as the parent cell. Cytokinesis. Okay. When you see cytokinesis, this is now the end part of the telophase or telophase 1. It is now, you can actually observe some cleavage or cleave or furrow in the cell. Okay, this is now your telophase 1 or your meiosis 1. Okay, as you can observe in the prophase 1, can you see my shared screen, my illustration? Grade 8.
Yes, sir.
Yes, sir.
Okay, so this is in the upper part of the illustration. This is the actual. Okay, this is the actual um, slides or actual specimens. You can actually see this one under the microscope. Okay, we gathered this one before, no? When I was still in college. The lower part are the graphical representation, and the actual slides are in the topmost. We actually created this one. Okay, if you're going to look at prophase 1, it is scattered. Oh, you look at scattered, but the centrioles are moving on the opposite sides. Okay, so the centrioles is in here, moving the opposite sides. Here, opposite sides here. Okay, now in metaphase 2, they are actually aligned in the equator. Now look at, this is the equatorial line. Okay, but they are not separated yet. Okay, if you're going to look at it, the spindle fibers are actually attached to the centromere, this one, and pulling it into the different sides or opposite sides. In the anaphase 1, you look at the arrangement of chromosomes in the anaphase 1. Okay, the centrioles are actually pulling it on the opposite side and leaving a cleave. So it will eventually create a cleave or cleavage. And then here comes telophase 1. Okay, two daughter cells, or in meiosis 1, we also two separated cells with haploid number of chromosomes. Okay, that's in the telophase 1. Okay, let's move now to meiosis 2, the equational division. When you say equational division, the daughter cells, which was created in the meiosis 1, will continue its journey to meiosis 2. Okay, meiosis 2 begins with prophase 2, whereby the nuclear membrane breaks up again and the spindle fibers form. Okay, the sister chromatids no longer replicated, but begin to migrate to the equatorial plate. In metaphase 2, sister chromatids line at the center of the equator. Okay, again, in every metaphase, there should be alignment in the equator, or else the cycle will not continue. It should be aligned at the center. This is to ensure that there is the equal or unequal sharing of genetic characteristic. Okay. In anaphase 2, the third part of meiosis 2, these sister chromatids split and move toward the opposite poles. Okay? And at the end of the telophase 2, a new nuclear envelope is formed or forms around the chromosomes. Nucleolus reappears and cytoplasm divides that and ends with four daughter cells, each with half number of chromosomes as the parent cell. Okay? So this is now your meiosis 2. Okay? Can you see the difference? This is the late telophase 1. Okay? When it starts again to move in the opposite poles, when it starts to move, um, when the centrioles starts to move again in the opposite poles, prophase 2 started also. Okay? Prophase 2 started also. And after which, it will align at the center. Sure, why it's 2? Okay? Remember your meiosis 1? Okay, your meiosis 1 produces 2 daughter cells. So these are the 2 daughter cells. Okay? And again, once the centrioles moves to the opposite direction, tendency is it will now start the prophase 1, prophase 2. Okay? And once again, the, the, the chromosomes will align at the equator, it will now start the metaphase 2. Okay, this is the metaphase 2. Okay? Once the chromosomes are being pulled by these centrioles and separates the sister chromatids, it is not chromosome anymore because it is um, pulled by the centrioles to form sister chromatids. It will actually form, or it will, anaphase is happening now, anaphase 2 is happening now because of this pulling of sister chromatids. And once this separates or creates a cleavage or cleave or furrow and forms a new membrane or cellular membrane, it will create 4 daughter cells in telophase 2. And again, the end of telophase 2 is the cytokinesis, the creation of new daughter cells or new cells with haploid number of chromosomes. Do you have questions so far?
No, sir.
Are you sure? Because if this is face to face,
Yes, sir.
you will have a lot of activities actually. Okay? But since then, I will try my best to give you an illustration of what is really happening in the cell under these cycles. Okay? This is really interesting. What makes this interesting is this is how we actually get the genetic materials from our parents. And this is how nature works. This is how God created us. That is why. And orientation, until now, it is not actually explained by science. What makes it oriented to the equatorial line? What makes the homologous pairs um, share these character traits? It is by means of God. There's no science that can explain that one. That is why until now, we cannot actually fully uh, identify the factors that these chromosomes shared these genetic informations. But we can identify their movements. These are the movements, right? These are the movements of chromosomes. But in sharing with the genetic characteristic, it's a different thing. And it cannot be explained by science. Even though we have genetic engineering already. In the genetic engineering, we can create our own genes, we can change we have gene splicing. We can change a particular gene. Like for example, if you want a baby to have a blue eyes, you can do that one by genetic engineering and gene splicing. But the sharing of this genetic characteristic, it is a mystery until now. This is really interesting, and I want you to continue doing the research with this. I want you to continue enjoying the topic itself. And this is not hard to tell you honestly, because we're just identifying the movements of these chromosomes. We're just identifying how these cells move, right? The illustrations, by the way, in the upper part is the actual specimen under the microscope. Okay? We really tried our best to capture every moment when I was still in college. That is why. Is it really amazing? Grade 8?
Yes, sir.
And hopefully also, um, along the way, if it is really, we can have the normal classes or the pandemic will um the pandemic will end, I will let you see the actual specimens. I have here the actual specimens. I will let you see how these can be seen under the microscope, because this is really amazing. Let's move forward. Spermatogenesis and oogenesis, the one that I'm telling you a while ago. Spermatogenesis is the formation again of sperms, and oogenesis is the formation of egg. Spermatogenesis started from oogonium, spermatogonium. Spermatogonium, this is a cell which we, or this is the parent cell of sperms, okay, before they actually undergo meiosis 1 and meiosis 2. Okay, in meiosis 1, we're actually creating 2 daughter cells, and in meiosis 2, we are actually creating 4 daughter cells or sperms. How about in oogonium or formation of egg? Okay, in meiosis 1, we are actually creating 2 daughter cells. Okay, the secondary oocyte, and the second one is the polar body. But in meiosis 2, we are actually creating 4, 1 oocyte or oogonium or ovum or egg, and 3 polar bodies. And eventually, these polar bodies will disintegrate, leaving only 1. Okay? At the end, actually, it's 4. But because we need only 1 egg per cycle, tendency is these 3 polar bodies will eventually disintegrate, leaving only 1 egg or ovum. Chromosome structure. Okay? Chromosomes are organized structures located in the nucleus of the cells. They carry information essential to cellular functions, such as cell division and cell differentiation. The term chromosomes comes from the Greek word chroma, which means color, or soma, which means body, a body with color, because we cannot actually see these chromosomes if it is not stained. Chromosomes are called such because they are strongly stained by some colorful dyes used in research. We usually put colors or dyes to it in order for these to be seen under the microscope, because these are highly transparent. Okay? Chromosomes appear double-stranded in the nucleus of the cell before division or cell division. Each strand is called chromatid, or we are putting a term sister chromatids. Is made up of a long molecule coiled around a protein called histones. The DNA molecule wrapped around a protein core called nucleosome. The nucleosome is the basic unit of chromosome. Chromatids are linked to a point called centromere at the center, which divides the chromosome into two sections or arms. The short section of the chromosome is called the P arm, and the long section is called the Q arm. So this is now the sections of the chromosome. Okay, this is where your um your centromere or your centrioles actually attach its spindle fibers. Okay, this is the centromere. This is your sister chromatids. Okay, this is the short P arm and Q arm. Telomeres, what are the telomeres, chair? This is the protection of the chromosome, not to be damaged. This is the end of your DNA structure. Okay. DNA structure and function. DNA is made up of many nucleotide molecules because the basic unit of DNA is the nucleotide linked to a long chain. The basic unit of DNA is called the nucleotide. Correct. Each nucleotide is made up of the following components. A 5-carbon sugar called deoxyribose. It's a sugar. A phosphate group and a nitrogenous base pair. Okay. The nitrogen-containing bases or nitrogenous base pairs are grouped into purine or purine, which include guanine and adenine. Okay. Purine or purine, it is adenine and guanine. Okay. Pyrimidines or pyrimidines are cytosine and thymine. How can we pair them? Okay. The DNA molecule is made up of two complementary polynucleotides. They are said to be complementary because the nitrogen-containing bases of one chain are bonded to those of the other chain according to the rule of pairing. Adenine is always paired with thymine. A is to T. Okay. Cytosine is always paired with guanine. C is to G. Hence, adenine and thymine are called complementary bases, and cytosine and guanine are also called complementary bases. So they're always paired. Okay. So hence the DNA structure. As you can see, adenine is always paired with thymine, and cytosine is always paired with guanine, and so on. Right? Gene structure. Do you have any questions so far?
No, sir.
A gene consists of two polynucleotide chains. One of the chain is called the template